In 2013, Lopez-Oding et al. Published what became the most cited paper in aging biology, The Hallmarks of Aging in Cell, identifying 9 fundamental molecular processes that drive aging across species. In 2023, the same team published an updated version in Cell expanding the list to 12. These hallmarks are not separate problems, they are interconnected processes that reinforce each other. More importantly, each one has documented interventions that slow its progression. This is the current state of the science, and what it means for anyone who wants to act on it. Lopez-Oding et al.'s 2023 updated Cell paper expanded the hallmarks to 12, integrating discoveries from the prior decade into a unified framework. The 12 hallmarks now span epigenetic alterations, telomere attrition, proteostasis loss, disabled macrophagy, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, altered intercellular communication, chronic inflammation, gut dysbiosis, and disabled autophagy, each with distinct intervention targets. This video draws from 5 peer-reviewed studies across Cell, Nature, and Journal of Internal Medicine, covering the 2023 hallmarks of aging framework, telomere biology, autophagy and aging, senolytic drugs, and dietary interventions in longevity. The traditional view of aging as an inevitable, genetically programmed process is being replaced, in the peer-reviewed literature, by the recognition that aging is a malleable collection of molecular dysfunctions, each amenable to targeted intervention. The hallmarks framework is not theoretical. FDA trials of senolytic drugs are underway, caloric restriction mimetics are in clinical use, and epigenetic reprogramming has extended lifespan in animal models. The science has moved from aging happens to aging has mechanisms, and mechanisms have targets. Cellular senescence, one of the 12 hallmarks, has emerged as a particularly tractable intervention target. Senescent cells accumulate with age, secreting a pro-inflammatory SASP, senescence-associated secretory phenotype, that damages neighboring cells and accelerates all other aging hallmarks. Kirkland and Shkonia's 2020 Journal of Internal Medicine review documented that senolytic drugs, dasatinib plus quercetin, navitoclax, Fisetin, selectively eliminate senescent cells in animal models, producing dramatic health span extension, with first human trials showing safety signals. The 12 hallmarks can be grouped into 3 tiers: primary, causes of damage; antagonistic, responses that initially protect but eventually become harmful; and integrative, collective outcomes. Understanding this hierarchy reveals the logic of intervention sequencing. Addressing primary hallmarks earlier is more effective than managing integrative hallmarks later. Fontana and Partridge's 2015 Cell Review synthesized the evidence for dietary interventions in longevity from model organisms through emerging human data. Their analysis confirmed that caloric restriction, reducing calories by 20 to 40% without malnutrition, is the most robust and reproducible longevity intervention across all studied species, operating through multiple simultaneous hallmark mechanisms. Telomere attrition, the progressive shortening of chromosome ends with each cell division, is one of the most studied aging hallmarks. Blackburn's research established telomerase as the enzyme that replenishes telomere length, and that telomere length in immune cells is a reliable biomarker of biological age and stress exposure. The 12 hallmarks of aging each have interventions with varying levels of evidence and practical accessibility. This ranking compares current interventions by their ability to address multiple hallmarks simultaneously. The 12 hallmarks are interconnected through amplifying feedback loops. Each hallmark worsens the others. This interconnection is also the reason that multi-targeted interventions, like caloric restriction and exercise, outperform single-target approaches. The evidence-based anti-aging framework targets the highest leverage hallmarks with the most accessible interventions, prioritizing caloric restriction mimetics, autophagy activation, exercise, and anti-senescence dietary compounds that collectively address 8 to 10 hallmarks simultaneously. Anti-aging interventions have different timescales of action. Some produce measurable biomarker changes within weeks, while others require years of consistent application to show detectable effects. The daily implementation of a multi-hallmark anti-aging protocol integrates the interventions with the broadest hallmark coverage into 6 consistent actions. The anti-aging protocol should be personalized based on age, health status, and specific hallmarks showing the most advancement in each individual. Aging has 12 interconnected molecular mechanisms, and each has documented intervention targets. Here is what the 2023 Cell Framework and supporting research show. The evidence is clear. The mechanisms are understood. And the steps to target multiple aging hallmarks simultaneously are within reach, for anyone who applies the science consistently. Follow MedBrief Today for more science-backed health insights, explained clearly.